So this question right here is kind of like big, but I'll read the question and I'll tell you everything. I'll tell you guys a simple way of trying to answer this, okay? So the question is you should state out the functional significance of cytoplasmic staining affinities of what basophilic erythroblast, polychromatic erythroblast, normoblast, and erythrocyte, okay? So one thing is, before you can be able to answer this question, first of all, they are saying you should state out the functional significance of their staining. So they are trying to you should explain if you stain a basophilic erythroblast, how will it look like? If you stain a polychromatic erythroblast, how will it look like? And how is the structure related to the function? Okay, so your knowledge of what erythropoiesis, that's the process of red blood cell formation, is tested right here. So you must know the functions of basophilic erythroblast. You must know their characteristics. You must know the characteristics of what polychromatic erythroblast and everything. So it might not be clear, but when we go in and start answering the questions, it will shed more light on it. So this is the process of what erythropoiesis. They asked you about basophilic erythroblast, polychromatic erythroblast. Then what is the other one? Normoblast. They also asked you about what erythrocyte. Okay, this is normoblast, also known as what autochromatic erythroblast. So, you must know erythropoiesis before you can answer this question, which is the process of red blood cell formation. If you stain a basophilic erythroblast, this is how it will look like. So why is it looking like this? You have to explain. If you stain a polychromatic erythroblast, it will look like this. Why is it looking like this? If you stain an autochromatic erythroblast, also known as a normoblast, it will look like this. Why? Okay, so I think this question in that exam was strictly for the distinction candidates because few people can reason sense out of this like this. So the cytoplasmic staining affinities of the cells reflect their functional significance. So how they stain reflects what? How they function. Now, if you stain a basophilic erythroblast, this is it. You'll be having a deep blue staining, and this is due to what? High RNA content. Do you understand? This indicates that what protein activity, the proteins is being synthesized by RNA and all that. So if it's having a deep blue staining, that means it has what? High RNA content. So that indicates that what? There's active protein activities in this type of cells. Okay, so the functions is what? They produce hemoglobin and other protein which is essential for red blood cell maturation. You are done. Moving to the next one, polychromatic erythroblast. If you stain it, this is how it will look like. Why is it looking like this? It is a mixed blue and pink staining due to increased RNA, and now there's what? Increased hemoglobin concentration. So the blue staining is due to what? RNA, while pink is due to what? Hemoglobin. Okay, this indicates transition from active protein synthesis to hemoglobin accumulation, because as the red cell is maturating, as it is what? Maturing, there's increments in one hemoglobin concentration. Okay, so that's why I have a mixed staining here. Function: it accumulates hemoglobin, preparing for erythrocyte maturation. Then next, we are talking about the autochromatic erythroblast, also known as normoblast. If you stain it, this is how it will look like. Now can you see that it is almost looking pink, total pink. So pink staining, it will show pink staining and a low blue. Okay, now it is showing pink staining due to what? High hemoglobin concentration, and the RNA is now reducing. Okay, it indicates nearly complete hemoglobin synthesis and maturation. Function is it's included in the final stage of erythrocyte maturation and preparation for release into circulation, right? Then last one, which is the matured red blood cell known as what? Erythrocyte. It's completely looking pink, right? This has a uniform pink staining due to high hemoglobin content. Now this indicates what? Complete maturation and lack of RNA. Okay. Function: since it is having hemoglobin, what is the function of hemoglobin? Transport of oxygen. Okay, so oxygen transport and delivery to tissues is quite high at this stage. Okay, so I think, and if you notice, the things we were talking about was just simply RNA concentration and hemoglobin concentration, how it is moving. But you can't answer this question if you don't know this what? Root. This is erythropoiesis. Okay, so that's why I always advise new students not to just jump to past questions. At least you can read handouts for a while. Let your head mature. Okay, not that you just come straight up and be cramming past questions like that. So just make sure you read erythropoiesis, everything before you try answering these questions. So that's it.